Okay, welcome to day 8, and today is going to be all about, actual, I guess, spillover and mapping it and things like that. And we're really, really excited to have Cliff with us, Dr. Clifton McKee. He's a research associate at the Department of Epidemiology in the School of Public Health in Johns Hopkins. He did his PhD at Colorado State University and then joined Johns Hopkins. He's worked a lot on Bartonella, which is kind of a nice pathogen because of its high prevalence, and it's a very generalist kind of thing, sort of like coronavirus in some sense. But then has been working more recently a lot more on Nipah, and that's very different in flavor in some sense, much more specialized. So great to have you, Cliff, and look forward to your talk.
Thank you. Thanks for having me. Thanks for having me out here. I'm really excited to be here. This is my first time in India. So it's been exciting to learn from all of you and participate in this workshop. So what I've seen so far has been very successful. So great job, Ansil and Uma, for organizing it. Okay, so well, my task for today is to talk about the linkage between stress and pathogen shedding and what we know about that linkage and how to study sort of these processes that connect the organism to the landscape and shedding and pathogen spillover. So I really have to give a lot of credit to the colleagues that I work with. So I'm part of a network of scientists called Bat1 Health, headed by Raina Plowright at Cornell University. So I lean very heavily on her work on Hendra virus in Australia, because it's just such a great example system to illustrate all of this. So I'll present a lot of kind of Raina's work here, but I have to give credit also to two other colleagues, Manuel Ruiz Aravena, who's at Mississippi State University. He sent me some slides for this presentation, and then Dan Becker at University of Oklahoma, who also kind of contributed to this presentation remotely. So thanks to them. And I'll start. I like to do outlines just because it gets my thoughts in order. So the structure of what I'll kind of go through today is how we link stress to pathogen spillover. I'll then give sort of a background on the effects of stress on the immune system. And then I'll go through some examples of how stress and land use change are affecting the Hendra virus system and leading to these spillover events. And then, potentially I wanted to give you all something maybe that you could take away from this presentation, in your own work. So I kind of just wanted to discuss some potential measurements of stress in the field and some thoughts about study design in case you're interested in this and want to kind of put it into your own work. Okay, so we've seen this diagram or some flavor of this diagram multiple times in this workshop, but it is really a great way of sort of organizing our thoughts here. And spillover is a cascade of multiple processes that have to occur, right? So you start with, where are your wildlife hosts? Where are they in the landscape? What is their abundance? Are they susceptible to infection? If they are infected, how intense is that infection? Do they have high prevalence, or are individuals, do they have very high pathogen loads? And then there's this process of when do they release these pathogens into the environment? And so, you can simplify that as like infection and shedding. Once you've kind of reached that, this is really the barrier, from green to red here is the barrier from the ecosystem, right? The hosts in the ecosystem to the recipient. Who is the new host that is going to be receiving this pathogen? And so this is really the interface, the human-animal interface, or wild animal, domestic animal interface here. And so that really depends on, does the pathogen survive outside of the host? How are humans getting exposed to it through behavior or resource consumption or anything like that? And then, is the human actually susceptible to this infection? And that really determines whether you get, an actual realized spillover event and then potentially spreading as well. So there's this nice cascade, and you can study spillover at kind of any one of these things, but ideally you want to study it as a cascade and study all parts of it at once. But that can be very challenging. But kind of how we see the pathogen spillover evolving is, on these landscapes, there's sort of like the Swiss cheese analogy that we often use, right? So you may have some hosts that are very widely distributed, and they're shedding, an infection into the environment. But there's no one there. There's no one there to receive it, right? They are shedding virus into an empty environment, so the risk of spillover is very low. But once you have, these changes to the environment that place new things into the landscape, you can create these opportunities for changing host behavior, right? So maybe they've lost some of their historic habitat, and so they have to move to new areas. So that might place them into proximity to humans. And then humans may also be placing things in the environment, right, that are vectors for spillover or are opportunities for spillover. And so we can think of stress happening on multiple scales here in this cascade, right? So this is a really nice paper by Reina and Lancet Public Health, and I'll walk through this table because I think it's a really great illustration of kind of this linkage between land use and stress and spillover. So they speak a lot about kind of landscape immunity, and you can think of landscape immunity as being sort of the collective resistance of an ecosystem to resist pathogen spillover through the integrity of ecosystems, all the species are there and are healthy, as well as the health of human populations. So anything that is going to help to retain sort of these integrated communities and everybody's getting enough food, just to sustain most of their life functions and all of their behavior, then they're going to be good to, they're going to be able to resist pathogens. So you can think of land use, habitat fragmentation and degradation and augmentation may cause disturbance to wildlife that cause them to move to new areas, right? So they might move from native habitats to disturbed areas because they've lost, this resource, and so they have to go find new things. So that's a level of a stressor. You could also have sort of changes to the ecological structure. You may not lose the whole habitat, but you may lose, prey species or keystone species in the environment that are holding things together, or important processes, right? So maybe something about the hydrology or certain cycles have changed enough that you can no longer stay there or have to go seek out these resources elsewhere. And so you can get sort of changes in food webs. At this next level, sort of at the infection stage of things, if you have this decreased natural resources, you may lead these reservoir hosts to go into a state of kind of nutritional imbalance, or they're running on empty, right? Because they can't gain all of the resources that they need to do all the behaviors they need or fight the pathogens that they're exposed to. You can also maybe get exposed to biological toxicants in the environment, that could be a change that exists that may decrease your immune function as well. And then also these changes in movement may change sort of the overall density of your population. And so you can have increased crowding, which may increase the transmission process, especially if it is maybe a directly transmitted disease, right? So you would have these direct interactions that are density dependent that might increase pathogen prevalence. And then, you have additional things that kind of can happen on the other side, like the recipient side of pathogen exposure, changes to human behavior, right? In this process of land use change, they may be doing things on that landscape that may expose them to pathogens that they would not have been exposed to before. But you can also get changes in human population health as well, right? So living in sort of these disturbed environments as well could decrease the health of human populations. So that may lead to pre-existing conditions like asthma or something that might lead to higher susceptibility for respiratory pathogens, or your own sort of nutritional stress and decreased immune function. So these stressors that are introduced from land use change and habitat degradation can affect all parts of this cascade. And we can think of sort of land use change and pathogen shedding. The effects of stress kind of may depend on the pathogen transmission mode, right? So you might have, it's hard to read this, I'm sorry if the text is kind of small, but it says here at the top, human modified landscapes and landscape change processes like habitat loss, habitat fragmentation, matrix intensification, like increasing the edge of habitats in a landscape. The effect of those land use changes may change disease prevalence for different transmission modes a little bit differently, right? So for like vector-borne transmission, you might expect that land use change may increase vector density. You might also lead to like low host species richness. So you might end up with fewer host species there, and maybe more of those species, a higher proportion of those species are susceptible to infection. It may contribute more to pathogen maintenance, sort of like the dilution hypothesis. And you also might have increased contact rates, either between hosts or between hosts and vectors, right? A host living in that environment may encounter more vectors in a degraded landscape. With direct transmission, you obviously might see increased contact rates, right? As hosts clump around limited resources, right? So if you degrade a landscape, the remaining resources, hosts may sort of gravitate towards that and as the only available place for them. And so overall in the landscape, those patches are very densely populated. And so that may increase transmission. And you also some species may respond very well to that disturbed landscape. Certain, rodent species may love to live in, degraded landscapes and may do very well there, and may contribute disproportionately to pathogen shedding. So that can help. Sexually transmitted diseases, kind of similar to direct transmission, you have increased contact rates that can happen. But stress, can also affect each one of these in different ways. So, the physiological stress response may increase, an individual's susceptibility to infection, which may also increase transmission. And there, a bunch of other different factors may come into play here, trying to link land use change to stress to pathogen shedding. certain biological features of the pathogen may be really important, including like its virulence, like I said, the mode of transmission, the host. There's lots of other host factors that can kind of determine how stress may play out in this environment. And obviously, like the environment depends on how connected the environment is at the beginning, and how much of that connectivity remains after disturbance. And then some of these other kind of extrinsic factors like climate and extreme weather events can really play a big role here. Okay, that's very general kind of linkage of all of these things together. I can pause for questions if anybody. I can have a sip of coffee too. Okay, so the next part is really trying to get into the biology of stress and how it links to pathogen shedding and immunity. So most of what we think of as the physiological response to stress is mediated by glucocorticoid hormones, so things like cortisol. And so in response to some physiological stress or even a perceived stress, so like staying up all night and studying for an exam 3 nights in a row can be a stress as well. So if any of those things sort of reach this level of being perceived as a stressor, it actually activates I'm going to butcher this pronunciation, the apothalamic pituitary adrenal axis. So basically it's this cascade of signaling from the hypothalamus to the pituitary gland that leads to the production of cortisol. So once you have cortisol circulating in the system, it can actually have a lot of these important effects on the immune system. So high cortisol levels can inhibit white blood cell activation and promote apoptosis of white blood cells. And at very high concentrations, glucocorticoids also inhibit the production of B and T cells. And they also, in a way, they also inhibit inflammation by dampening the signal transduction downstream. So it has all of these kind of multiple effects on the immune system. And those are important for just regular functioning of the immune system, because these hormones sort of act as like the brake pedal on your immune system, right? So they can help with early activation. So they can increase these these PRRs, these things that are recognizing pathogens in cells and responding to it. It can help increase cytokine receptors and complement sort of these innate immune responses. So having glucocorticoids there can help make sure that the immune response, the innate immune response, is happening very quickly. But they're important later on for turning off these pro-inflammatory cytokines and chemokines, and also changing sort of like antigen presentation, T cell receptors and lymphocytes. So they upregulate the expression of genes that are involved in the detection of pathogens in tissue trauma, including pattern recognition receptors, these PRRs, cytokine receptors and complement factors, while inhibiting expression of these pro-inflammatory cytokines. So it has actually this sort of, it regulates the immune system sort of in this biphasic manner, such that at low doses, things like cortisol promote the expression of innate immune genes and rapid responses to stressors, pathogens that are there. But if you have stress that like really prolonged stress, chronic stressors, or in the case of people who receive steroid hormones as a treatment, sort of like pharmacological doses of glucocorticoids, they can actually suppress the signaling. And so you can actually get this effect where you can have, you may not be able to mount a very strong immune response. So backing up a little bit, what do you all think is stress? What would it, for an animal living out in an ecosystem, what is a stressor that they might experience?
I think that's a really good question because we use stress very colloquially also, right?
Yeah.
So yeah. Yeah. Anyone?
I very much, yeah. Go ahead and repeat the question. Yeah.
So I was just saying, stress could also be very subjective and species specific. What we deem as stress for one species not necessarily means it's stressful for another species.
Yeah, like so a species that lives in a temperate zone may have, a large snowfall event or a very deep freeze event, maybe a stressful environment, like event for them. But a penguin living in Antarctica, snow is very normal, and actually the absence of snow may be more of a stressor to them.
Stress also happens at different timescales. Like there could be something that happens at a longer time scale, and if you just focus on a very short time period, you might not see it as a stress, but only if you look at it from a big picture, larger time scale, you will understand that something is happening. So,
Can you give it an example?
like climate.
Okay.
How it acts on animals may not be like immediate, unlike some weather events, like some heat waves. It happens in few days, whereas some other climate processes that happens at a larger time scale, so you might not see it immediately, but over time only you will see the effect of it. So
Yeah, no, that's great.
Availability of shortage of resources
Shortage of resources? Yeah.
or elimination of one source in that given environment. Say, for example, application of pesticides or insecticides or destruction of forest in particular area, then that makes that population under stress because of lack of resource. Food, for example, food. Then that may increase the stress in that population of that area.
Yeah, no, that's great. So yeah, resource restriction.
So what I think, like all living things are evolved their own optimum conditions. So when they are out of the range of their optimum condition, they feel stress.
It could also be anthropogenic disturbances in landscapes. For example, for bats, for that matter, if people are going continuously to a roost during the daytime when they are roosting and disturbing them, that could affect with the physiological functions, and that could also cause stress.
And things like hunting or harassment of species are just kind of another level of that, right? So those are really great. I just have a few other examples, sort of, predation, right? Can be a stressor for a prey species. Interspecific crowding, so like, if conditions change where there's just way too many of you in a small area and not enough resources, you may have increased aggressive responses intraspecific aggressive responses. In very social species, change in the dominance hierarchy can be very stressful. So like in chickens, for example, there's the pecking order. So if you have changed, your dominance hierarchy, the ones who are lower in the hierarchy can experience lots of stress from that change. For species that mate, with a single mate for life or for a long period, the loss of a mate can be extremely stressful. Your point about inadequate nutrition, dehydration, extreme weather, those are all things. But also, just energetically taxing biological activities. So pregnancy, birth, and lactation are all stressors that an organism experiences. And so, whether it counts as a stress, whether, something really causes sort of this aversive stimulus, depends partly on its duration, and whether it's really part of an expected, it's an expected part of an animal's life. So something like pregnancy, birth, and lactation, those are expected things. Like in some way, an individual is prepared for that. But whether that stressor is layered on top of additional stressors that might be in the environment may lead to sort of this aversive response. Yeah, aging, that's a great one. And then co-infections, right? That's the other one, right? You may have, a very ubiquitous parasite that exists in the environment, and then suddenly a virus comes in, and that's layered on top of this other co-infection that you have. So that can lead to stress. So this kind of the dependence of stress and its effects on the immune system does really depend on this duration of the stressor. So something like an aggressive interaction or a very temporary parasite or predation, those are things that happen on like minutes to hours. And so they can lead to a spike in cortisol, right? As a way of sort of preparing the immune system for this insult. And so you may, see an increase in basal immune function, right, as animals prepare for this. And that's a good thing. Like that's actually really what the immune system is sort of, or this stress response has evolved for, right, to preempt some of these insults. And things like, inclement weather, a very rainy period or a big snowstorm or social status change or food restriction, something that's, maybe lasts days to weeks, that may be something that an animal can get through just fine, right? They've enhanced their immune system, they're prepared for this, but it's when it actually starts to get into this really prolonged, stressful situations, like habitat modification that has, gotten rid of all of these, the necessary resources for your life, or pollutants, right, that are very persistently in your environment, and other like invasive species, right? That's another one like that can change aggressive encounters or predation, or they may be eating up all of the things you normally eat, or taking up all of your normal roosting areas. And so these things, it's when these stressors start to become prolonged and chronic, we actually see a change in the immune system to a phase of sort of immune suppression. So, these short-term insults to, short-term stressors can affect some different arms of the immune system. So you can have, short-term things that increase sort of this inflammation, this innate immune response that's, detecting the pathogen and responding to it with phagocytosis and all these other things. And then as these stressors sort of prolong, you may also get things like elevation of T cells and B cells, but it's only at, a really long, chronic, stressful event that you actually start to hear this, increase in immune function and then a start to decrease. And actually you get sort of this bifurcation in the inflammation response, where you may actually end up with chronic inflammation that can lead to sort of more pathology that's happening in an animal. So there's a couple of concepts that get used in the literature when talking about stress and its relationship with the immune system. And those are allostasis and allostatic load. So allostasis is really like, it's the analog of homeostasis, right? Where an animal is doing everything to regulate these internal processes. But allostasis is sort of finding stability as an organism through change, through adaptation. So it's sort of extending this concept of homeostasis in a world where things are changing all the time. Either predictably, right? So animals are responding to seasonal changes in resources, or unpredictably, right? So changing in human disturbance or just other changes in the landscape that are not expected in their lifetime. And that you can take this concept of allostasis and you can kind of think about allostatic load as being the total resources that an animal will require at any given time to do the things that it needs to do, right? So that includes metabolism, just growth and repair, thermoregulation, any of the behaviors that it normally does in terms of foraging, mating, and other reproductive activities. So it's sort of the well of resources that an animal has at its disposal to do the things it needs to do. But you might reach a level of what we call allostatic overload. And that's sort of this state where the cumulative resources available to an individual are exceeded by the sum of its energetic costs, right? It's reached this point where the demands of energy on an organism are too much for what it can consume in a day.
So the allostatic overload may also lead to the stress?
I would imagine the stressors are the ones that are causing the allostatic overload. And I'll get to it. I have a nice diagram to kind of illustrate this. So imagine, there's this energy budget that an animal has. And so this green curve is showing the energy demands that an animal has just to exist daily. So just metabolism, feeding, foraging, any of those things. Those may change over time, right? Like throughout a year, you may actually have, you may be able to get more energy because you can forage more, there's more resources in your environment, and then during periods like winter, right? There's less available for you in the environment. You also have these other activities that you might have to do. Oh, sorry. So this is the demands. So green is like what you need to do. The purple stuff is some other additional things that you might do in your life, right? So mating, migration, for birds, molting is like a very energetically taxing activity. So those may happen during certain periods of the year where there's enough food for them. And animals have optimized their life history strategies based on the energy available to them in the environment. So that's kind of what we're showing here. So imagine like the buffer, this blue bar is like how much they can forage, right? So this is the total energy that's available to them. And so animals are sort of optimizing when to do some of these energetically taxing activities to the times when they have enough energy to do them. But, so for bats, like pregnancy and lactation is a pretty stressful activity. Like being a bat itself is pretty taxing, right? So you're a warm-blooded organism, you're flying, and then you have to carry around a live pup and produce milk for it. So during this period of lactation and carrying around these pups can be really energetically taxing for a bat. So stressors can begin to change this energy budget, right? So say land use change actually has decreased the amount of things that are in the environment, like maybe this blue bar has come down, or something has changed in the environment that it increases the daily the daily cost of just being in that environment. Say, for instance, they've clear-cut the forest, right, that you normally foraged in. And so you actually have to fly further to actually get all the food. So maybe your total energy budget maybe didn't change too much, but the cost of acquiring those resources increases. And so you no longer have this like nice little buffer where you had surplus energy available to you in the environment. So animals here may be sort of living on this edge. If, say for instance, this stressor, this modification has become so pronounced that the total of, this blue buffer, these resources available to you in the environment have just plummeted. You just have nothing there for you. And so these activities that you normally do that are part of your life history still need to happen, but now they're happening at, in a place where you don't have sufficient energy to do them. And so that can be a very stressful, physiologically stressful scenario for an animal. So that's the idea of allostatic overload is these animals are trying to live their life, and they are expecting a certain availability of resources, and suddenly those might disappear. And the effects of stress may really depend on the pathogen itself and how it's transmitted. So I don't know if we've covered a lot of this so far, like these concepts of different transmission modes or disease dynamics. Have we covered it? Okay. So one of the ways we conceptualize disease transmission in organisms is through these compartmental models, right? You can take one proportion of the population and you assume they're all susceptible. And you also might assume that another small proportion is infected. And basically you can simulate the transmission process and watch as individuals who start as susceptible become infected and then recover. And then you can see like individuals will, as they become infected, they mount an immune response, and then maybe are immune for the rest of their life. So like typically like an acutely immunizing infection, like measles, for instance, right, might follow a nice SIR dynamic, right, where everyone starts out as a baby as susceptible, they might get infected, and then once you're infected, you never get infected again. You're recovered and you're resistant. But some diseases, maybe your immune response may only be protective for a certain amount of time. So SARS-CoV-2 is a great example of that. Like your immune response wanes over time. And so you might start out as susceptible. In 2019, we all started out as susceptible to SARS-CoV-2. We've, pretty much all of us have been infected at some point, or have received a vaccine, and you recover. And so you're resistant to reinfection at that point. But that immune response may wane, and you may become susceptible again. And so the cycle can repeat, right? So you could get infected multiple times in your life. There's another kind of interesting dynamic called silly, which instead of recovered, you may not fully recover from infection. You may actually your immune system may only have a partial clearance of this infection, and it may go into a latent phase. So things like tuberculosis, a lot of herpes viruses, HIV to some degree goes through this. And so during this dynamic, right, you may experience infection really quickly, or it may go through the population very quickly, and then have this phase where it's kind of smoldering, maybe undetectable in you for a long time, and then it may recrudesce and show up again, and can become infected. Yeah.
What are the immunological markers to identify this kind of a dynamics? Like antibodies, which is it antibody, or what do we look at to identify this?
I think very typically it's they use IgG, like immunoglobulins, antibodies. So, during that very quick convalescent phase right after you're infected, you may have IgM, and then a few days later to a week later, you might start presenting IgG. And those are the protective ones that are neutralizing.
SILI.
Oh, for SILI, I don't really know. I think that one's hard. You'd have to look into the literature of, measles can have sort of this latent phase. And so it can actually hide out, I think, in the lysozyme and things there's lots of places where it can hide out. So I don't know a full answer to your question.
One more question is, we are able to see this at an individual level, like measure whatever metric for this kind of a dynamics. How do we course grain this to a population level and see how that particular blue curve, how do we see, how do we make assumptions about the dynamics about the population level by taking it from an individual level?
Yeah, I mean, there's a whole, realm of, disease ecology that touches on dynamical models. So I'm not going to go into it here. It's basically a system of partial differential equations. And so you can actually do the math and make some assumptions about the transmission process and the density dependence of transmission. So there's great courses online, like Coursera courses, that go through an introduction of that to take, to be able to model this at a population level. So yeah, and I can share links with you for those courses if you want.
I try to answer for his question, how we can trace these markers. As I said, the IgG, we can correlate the antibody titer for that pathogen.
Yep.
So antibody titer as well as pathogen load, simultaneously if you trace, we get this type of pattern. So in case of SILI, where it is for specific pathogens, say for example, if the pathogen enters, definitely IgG level goes up. So IgM and IgG antibodies are not the only protective molecules, but also many other cells are involved in the elimination of the pathogen. But here in SILI, sometimes the pathogen adapt better. It hides. Say for example, if the pathogen, it becomes intracellular, where there is that is not accessible for the antibody. So therefore, it will escape from antibody. So it will be within the part of the host cell. So whenever it waits for some time, when the stress is very high, so definitely again it triggers to its replication, amplification of that pathogen. Again, once it is come out, again the antibody play a role. Again it releases antibody. Again it controls the level of pathogen. So where it will be continuous and it is specific to each pathogen, type of pathogen within a given host. Yep. So I think.
Yeah, so it's hard to answer. I think it really depends on the organism, the pathogen that you're looking at.
This is stress response is after the pathogen attack or before the pathogen attack?
I'll just move to the next slide and then hopefully that'll answer your question.
No, I'm just asking to this. After the pathogen attack what you're telling that is this the stress response after the host is attacked by the pathogen or before the attack of pathogen? Immune response, right? If in that case, what happens in immune response means the IgG level won't increase actually. In the chronic stress immune responses, there will be inhibition in the immunity, right? So there won't be increase in any immunoglobulins in the host.
Wasn't that at a population?
You can also, these curves can be at an individual level and also a population level, right? You kind of see you can see these kind of increases in this scale might be individual pathogen virions, right, or bacteremia, but it also might be indicative of total infected individuals in a population. They can kind of follow these same things, right? Because if an individual mounts an immune response, becomes infected, mounts an immune response, and clears the infection, and then over time becomes susceptible again, you can see them getting infected again, and so you might see a rise. But the thing is, and this gets to your point, right? If you have repeated exposures to a pathogen, you may also you may actually increase the titer of IgG, right? So the second time you're exposed to it, you may not get it as infected, and so maybe bacteremia or viremia might be lower. But yeah, and at a population level, right? This could be total infected individuals and sort of the proportion of immune individuals in the population, and that might wane over, this decreases as individuals, the kind of collective population immunity wanes. And so you might have a repeated outbreak that happens. And so that's kind of the interesting thing that happens when you see kind of biannual phases of like diphtheria and things like that. So that's really at a population level, your population immunity can wane over time, especially as you increase, add new births into the population. Like new individuals born into the population are always susceptible, and so you're kind of adding this waning on top of introducing new susceptibles into the population. But this question of just like where does stress play into this role? Oh, go ahead.
So these are, if there are all population models, but with the new infectious diseases, when they start to spread, they are usually 2 to 3 individuals. Can we say which model will a pathogen might belong to at least? Can we know which model within those 3 the infectious disease can take?
We can make it. Usually you can make some assumptions about what it will do. But often when it's a very new pathogen, it's hard to tell. Because I think early on with COVID, I think we assumed it was SIR, right? That once you had it, you'd be fine. We didn't know that you could become susceptible again, that the immunity would wane so fast that it would actually become more something like an SIRS, right? Where you can be repeatedly infected. So you can make some initial assumptions, but biology will prove you wrong sometimes. But okay, so there's the text is small here, but what I'm showing is sort of like how these models might work in a population. So on the right-hand side, I have an SIRS, right? So you have individuals who are susceptible, and you might introduce a pathogen into that system here at the top. So an initial individual who is susceptible may be infected, and they become infectious, and they can start leading to transmission within their wider population, and they might get susceptible again, right, over time. There's all of these sort of extrinsic factors that can contribute to this transmission process, right? So contact rates are a really important thing. So how frequently or how often or how intensely an individual has contact with other individuals in the population, right? So the contact rate you have with your family might be way higher than the contact rate you have with a colleague, right? So you spend more time and you have closer interactions with those individuals. And the same thing might happen with bats, right? They might have closer interactions with a small set of individuals and may have very few interactions with a roost or a population that lives 10 kilometers away. It also depends, and these contact rates kind of depend on the availability of food, the distribution of that food in the environment, these environmental perturbations that would change their behavior and how animals go about their foraging, reproduction, right? So during most of the year, you might have some level of contact with some individuals, but during a mating season, that changes, right? You'll have greater interest-specific contacts. And so all of these kind of contribute to this transmission process. For both, SIRS, or these sort of latent, silly models. But when you introduce stress into this equation, right, that can really affect sort of this process of changing from a latent host to an infected host. So you may be a latent, the pathogen may be hiding out in some, it may be intracellular, it may be hiding out in your central nervous system or something like Borrelia does. And something might change in your environment or your physiology. You may go into nutritional stress, right? I'm not getting enough food, or I'm dehydrated. And so that can lead to this cascade of, this hormone cascade that can reduce your immune function, that's maybe keeping this latent infection at bay, at a very low level, so that it starts replicating more. And so you actually become infectious again. So stress can play a lot in here. It changes all of these, stress may change all of these other things, but that's the other part of the equation that's sort of unique to silly, is that stress can sort of directly affect whether you become infectious or not. Okay, more questions before I jump into Hendra. Yeah.
So density with loss of resources could be something that is stressful. So I'm just assuming for roost, like cave roosting bats which roost in high numbers together. It's only when a resource depletion or something happens that they are stressed. But in itself, like most of these colony roosting bats generally roost in high numbers, right?
Right, and that's maybe very normal for them, right? Like some species are very used to living in a colonial environment. But if something changes in their environment where there's increased, interspecific competition, or just more, there's a much larger increase in density than they've ever seen before, so that you have not only increased, contact rates, but many of those contacts may become more aggressive, right, as you fight for space. So I think in both of those ways, it can serve as a stressor, right? Because there's increased energetic demands. There's the perceived stress of having sort of like these aggressive responses, but there's also the increased contact rates that would increase, direct transmission of the pathogen. So in some ways, sort of density is affecting pathogen transmission through multiple, kind of directly and indirectly, right? Directly through density, but also indirectly through stress and immune suppression that you're seeing because of this stressor. Does that make sense? Okay.
Follow up. So I'm just for the species of bat that I work in called Latidens salimalis fruit bat. So they it's been observed that during monsoons they leave. So generally they are in like smaller roosts spread across the landscape in cave systems, and as soon as like the monsoon starts, and these are like riverine caves and places, so most of the caves gets flooded. So all the micro colonies come and stay as a super colony during monsoons. Could that also be something that could induce stress?
It could, but again, it may be very like species specific, right? Whether they perceive that as a stress, it's hard to tell, because that could be an expected event, and so that they're prepared for it. They have enough surplus of energy to kind of get through that stress, through that small period of time. So I think you would have to do some measurements of stress in that population to see whether they're kind of like they have lots of high levels of cortisol that are accumulating, and then is that affecting some immune parameters? so I think that's worth investigating, like whether they actually perceive this as a stress. Okay, so I'll dive into the stress and how it's affecting the Hendra virus system. So I don't know if, does everybody know what Hendra is? Have we covered it before? No? Okay. Okay, so Hendra is a zoonotic, bat-borne virus. So it's a paramyxovirus. The genus is Henipavirus. It was first discovered in 1994 during an outbreak of kind of mysterious disease in Australia that was killing horses. And it was causing 70% of mortality in infected horses. Initially, they thought it was a whole bunch of different things, but eventually they isolated this novel virus. And they found later on that the host of this virus is Pteropus species fruit bats. So there's 3 different species in Australia that contribute to being reservoirs of Hendra. And the way it spills over into horses is basically bats shed this virus in their urine. And it happens when bats are feeding on fruit resources that are on horse paddocks, right? So in Eastern Australia, there's been kind of this land use change that has, changed these native forests and kind of savanna habitats into agricultural land. And they've placed horses onto those landscapes. And so these bats, you have this overlap of this reservoir host and the intermediate host. And humans get infected through exposure to sick horses. So basically, the owners of the horses or veterinarians who are treating these sick horses are the ones that are getting infected. The number of human cases has never been that high. It's probably less than 100 at this point, but horses, there have been outbreaks. There's been dozens and dozens of outbreaks of Hendra in Australia since 1994. So once they recognized it, they kind of see it all the time. And this diagram that I was showing you before is really fit for Hendra, right? So basically, Hendra has probably existed in these bats for millions of years. And they were shedding this virus into the environment occasionally, but no one was there to receive it, right? So there were really no spillovers, I think, that were perceived at all. We didn't know anything about it. It was only as land use change in Australia had cut down some of these native forests that bats were living in and replaced that with agricultural land with these intermediate hosts in the form of horses. So these bats actually, in response to the land use change, moved more into these agricultural areas. And that proximity opened up this opportunity for spillover to happen. So something about the dynamic, like the general biology of Hendra virus is all of the shedding and all of the spillovers really seems to cluster in winter. So they've, like Reina and her team have been monitoring multiple populations of Pteropus bats in Eastern Australia since the early 2000s when they started doing this work. But this monitoring has been going on since 2012 is what I'm showing here. And so there's these multiple populations. And what this graph is showing is just the prevalence of, it's actually measuring like aliquots in roost urine. So they put out these tarps and they collect a bunch of roost urine from bats living in roost above it. And then they'll test a proportion of those samples. They'll like suck up a bit of the liquid and set up a bunch of aliquots and test those. So this is what they're showing here is the proportion of positive aliquots in those that are tested. And so across the board, this period here, this dashed line, is the winter period. And so most of this spillovers, most of the shedding is really concentrated during winter. And this is the austral winter, so it's in like June through August. And you also start to see sort of these larger magnitude shedding events in a winter that follows a food shortage. So these bats feed on nectar mainly during the spring, and they can tell from like really good record keeping in Australia about that apiaries actually take. So they use bees to pollinate a lot of the resources, a lot of the farms and crops in Australia. So they actually have good records of apiaries. And they can tell basically when there's a collapse of the nectar resources based on that. And so these kind of darker bars are showing the collapse of nectar resources. And so these big shedding events are actually happening in the winter following a food shortage. And the actual outbreaks that we see in horses are also concentrated in winter during this period. So there's a nice alignment of shedding and spillover. And these bats, you know, they can feed on nectar resources, but they can also feed on fruit. When these nectar-producing trees are available, they will fly super long distances to get there. And so this is just showing GPS tracks for individual bats going hundreds of kilometers to go feed on this nectar. But when there is no nectar, like the resource is not working for that year, they'll stick around more locally and try to feed on whatever they can. And that's often fairly suboptimal food. They actually go in, the classic example that Reina always gives is bats were actually going and feeding on lemon trees in some of these agricultural areas. So it's a really resource poor food. So they were kind of reaching this level of nutritional stress. And in Australia, you know, these pollen and nectar producing trees, it's mostly eucalyptus trees or gum trees. A lot of that habitat has been cleared. So this is like kind of what they think the resources looked like before Australia was colonized. And then like by 1996, a lot of that area had been gone. And then even more, 2018, a lot of those resources have been depleted. Yeah, Ansil. Oh, okay.
So about the movement pattern, won't you expect them to go look around for food a lot when there is low food at a particular location? Or is it something to do with energetics, like, you know, no food, so better not to go far or something like that?
I think that's part of it. They may try to just find whatever they can. The other thing that's sort of happening here, and I'll talk about it in a minute, is there's been this process, you know, historically these bats were doing that. They were flying over wide areas and just feeding on sort of these nectar resources as they became available. And they only clumped up and had these really large populations when there was sort of enough of a resource, a good flowering event at a given space and time. And so you would get these huge aggregations of like hundreds of thousands of bats all at once, and then they would spread out and go. What's happened is as they've lost those nectar resources, they've actually moved into agricultural areas. They've moved into more urbanized areas, and they're feeding on people's backyard trees. And so they don't have to go as far because this environment has sort of laid it out for them, but it may be suboptimal food, right? So they're trying to make do with what they can.
Is there a radio tag data? This one?
Yeah, this is all GPS. That's GPS data, so that you can actually put these little backpacks on the bats. So yeah, we've lost these nectar resources, and the other thing that's happened is, you know, these giant aggregations of bats around these nectar resources. You could see them still in the late 90s, but since about 2005, 2006. It's only happened once in 10 years that you see these huge aggregations. Most of these bats are living in living in smaller roosts. And this is kind of another way of displaying it, right? So this is just sort of a binned distribution of the roost sizes. So you have, the red is roosts of less than 450, the blue is anything over 25,000. And so you see the decrease over time of, the proportion of bats living in these really large roosts, those are declining, and you actually see an increase in sort of the proportion of bat colonies that are much smaller. So on the 450 count side. So yeah, these bats are sort of what we say is there's been a fissioning process, right? They used to live in these giant unified colonies, but they've actually spread out and they're no longer nomadic, they're more resident in these sort of backyard farms in agricultural areas. And that's kind of what I'm saying here. So as their winter habitat has been removed, right, they are moving into cities to just use those resources there. And that has an effect on Hendra shedding. So I already told you that shedding mostly happens in winter, but the intensity of that shedding really matters where they live, right? So these bats that actually live in this historic, overwintering habitat, this intact environment where they have access to these good nectar-producing trees. During winter, you only see maybe see a small increase in Hendra shedding. But in these urban roosts, these resident urban roosts, you see a much greater spike in winter. And then when you add on to that, right, these periods where there's a food shortage, right? So there's been a collapse of the nectar resources. The bats have to adapt and find some other things. It causes an increase in the shedding in both environments, right? Even in overwintering habitat, you'll see an increase in those years where there's a food shortage. But the magnitude of that increase is much larger in these habitats that are degraded. And the other thing that's happening is not only is, you can see this, the number of positives is increasing, right? So this is a measure of sort of like the number of aliquots under a roost that are positive. But you can actually, take a look at the CT value from like RT-PCR, and that gives you sort of a quantitation, like you can quantify how much virus was actually in that aliquot. And you actually end up seeing that like there's more virus also in those aliquots. It's not only just like the proportion of aliquots is higher, but there's more virus in those aliquots too. So they're having these really big shedding events during winter in these degraded habitats following a food shortage.
So, nectar sources, is it a direct correlation to the changing flowering patterns? And so many flowers are changing their seasonal patterns of flowering and all because of climate change. Is it a direct correlation to this?
I don't know. You're asking sort of about the phenology of the trees themselves. I don't know.
Okay.
Yeah, sorry. Probably. I think it is, and I'll talk a little bit about. There is an additional effect. There's a climate driver, I think, on those flowering events as well. So kind of what I'm talking about here is kind of these layering on of allostatic loads. So these multiple stressors that exist. So in winter they may be having there's higher thermoregulatory cost of just existing. You maybe have less food available to you because like some trees are not fruiting or the nectar is not producing. And then you also have to maybe fly longer distance to find all the food that you need to get. And so, these bats, when they're doing fine, right, when they have adequate energy, they're protected from pathogens. Right? So they may have an adequate immune response to like deal with this pathogen threat and maybe able to clear infections quite quickly. But during these kind of these sinks in their landscape of energy during winter, food stress and everything, that's when you see this pathogen, the really high pathogen shedding. So one of the things that they've actually been able to do with this system is take all of this data, this really, really dense data that they have about bats and Hendra virus shedding and spillover events and food shortages, and be able to link it all together into sort of this cascade effect of what are the effects of climate and food resources on pathogen shedding. And so what they've actually been able to do is show that there's a three-phase process. So first is this effect of El Nino. So El Nino has a really strong effect on Australian ecosystems. It really changes the prevailing temperatures and rainfall patterns. And so in years where there's a really high oceanic Nino index, and you have a food shortage that follows that in the spring. So there's an El Nino event, there's a food shortage, that is when you see spillover events, right? So it's first there's El Nino, then the bats experience this food stress, and then they go into winter, and then there's this big shedding event. And these food shortage events have had sort of this stepwise effect on the size of roosts. So back in the 90s, we had very few roosts in the environment, but they were just very large. But as each food shortage has happened in this landscape, these populations have fractioned, right? And they've broken up and joined these smaller roosts. And so we now have many, many more roosts in the environment. They're much closer to each other, but they're much smaller, right? So we've seen this stepwise effect on food shortages on these bats. So they're sort of perceiving that the signal from the environment is, we no longer can live here, we need to go somewhere else. And so they're going into these urban areas. And so you can kind of build this really great explanatory model for Hendra virus spillovers. So, 2 years, with a lag of 2 years back, right? If there is a high El Nino index, and then you have a food shortage, you'll get a spillover. And we can kind of, perceive these food shortages as these fissioned roosts, the increase in these smaller roosts. And you also see at the same time, these bats are feeding more in these agricultural lands. So all of these things are sort of contributing positively to the likelihood of spillover. And whether they're foraging in an agricultural area they actually have the highest probability of a spillover from a given roost, right? In forest and urban areas, there's a bit lower probability that there's going to be a spillover. So it's really these agricultural areas where they're intersecting with horses where you actually have the opportunity for spillover. But if you have these winter flowering pulses, right? So these gum trees and eucalyptus trees have these gigantic kind of masting flowering events that produce a ton of nectar. They don't exactly know what predicts those. They happen somewhat sporadically. There's not a great climate, El Nino doesn't actually drive that one directly as far as we know. But when they do happen, they actually are protective against spillovers. And the reason is because in one way, it alleviates the nutritional stress, right? So these bats have a great resource that they can go feed on. and prevent maybe this additional, the change on their immune function that might prevent them from, because we think as well in Hendra that maybe there is this sort of silly dynamic, right? So individuals may get exposed early on in life, they become infected, they clear the infection, and they become latent. And so, they may spend a lot of their life sort of latently infected, and it's only during those periods of stress that they actually recrudesce and have an additional infection. So having enough resources will prevent those individuals from recrudescing, right, is the hypothesis. But the other effect is sort of just on density. So these bats are no longer interacting, they're no longer on this agricultural area. They move away, they move back into the forests and are actually foraging away from agricultural area. So they're no longer there to interact with the horses. So there's a nice video here. I know I'm hopefully not running out of too much time. If I go over, is that okay a little bit? Okay. All right. Reina has made a really great video that explains this whole thing. So.
We studied the large nectar-feeding bats that are hosts of the Hendra virus, a virus that spills over from bats to horses, then from horses to humans. About 80% of the horses and 60% of the humans who contract the virus die, but bats are unaffected. Traditionally, these bats form large roosting aggregations that feed on nectar and pollen from ephemeral pulses of flowering in native forests. They migrate large distances to maintain access to a constant food supply and move pollen across the landscape, keeping forests healthy. However, every few years, the El Niño, Southern Oscillation Cycle, generates hot, dry conditions. The flowering of native trees is greatly reduced in the months following a strong El Niño, creating a food shortage for bats. The bats respond by splitting into smaller groups, close to poor quality food sources in human areas like cities and farms. In the past, all bats reverted back to migrating among pulses of native flowering once the effects of the drought were over. But recently, the splitting behaviour is persisting. An increasing proportion of bats are staying in small groups near farmland and cities all winter, eating fruit from weedy or ornamental trees. For example, trees planted for shade in horse paddocks. We think the reason they are staying is because we have removed their winter habitat. In winter, only a few types of trees reliably provide flowers and nectar in subtropical Australia. They grow primarily in coastal areas on land preferred for agriculture and housing. Most of this habitat has been cleared and continues to be cleared, reducing the bats' winter food supply. As a result, there are fewer big blossom events that bats can migrate to, which would have drawn them away from human areas. When their food supply is affected, the bats in human areas are stressed and hungry and start to excrete viruses in proximity to horses, who then die of the virus. We can predict these spillovers. Every strong El Niño year is followed by a food shortage for bats, and the food shortage is followed by a cluster of Hendra virus spillovers. However, that risk disappears when a substantial pulse of flowering occurs in a remaining patch of winter habitat. During these winter flowering pulses, bats leave urban and farm areas to feed in native forests. Every time that happened during our 25-year study, there were no spillover events. So, we think there is an obvious solution: protect and replant winter forests to address spillover at its root cause. We studied
Okay, so just generally, I wanted to talk like about stressors and like the cumulative stress. Oh, go ahead.
Also, these horses were not native to Australia, right? So most of the people, even the indigenous people, seem to be resistant to Hendra as compared to the people, the colonizers. Right?
I don't know if we can necessarily say that because there's never really been a direct spillover from a bat into humans. It's only ever been through horses, these intermediate hosts. So it's hard to say. I don't think anybody's studied the indigenous populations there very much.
And no native animals are known to be hosts. Other native mammals have been known to be hosts or intermediate hosts.
I think when they tested, initially they tested all types of animals to figure out who the reservoir host was, and they didn't find anything. You do see occasionally that it will spill over also into cats as well. Like there have been individual cats who got infected during these outbreaks, but that's it. I don't think there's like the native like marsupial populations, I don't think have ever had it. But there was one study that actually kind of looked at cortisol in these bats. They actually were able to measure urinary cortisol. Oh, go ahead.
So about the flowering example, does it also depend on the amount of flowering? Because there's a trade-off between what radius you can forage and amount of resource that you have. So if you have more flowering resource, you can forage more areas and come into human habited areas and then come into contact with these horses or something. And if you are below a threshold, then you don't have enough energy to forage a larger radius, so you don't come into contact. So does it depend on the context of how the flowering happens?
It does, and I think most of the flowering is happening away from agricultural areas anyway. And so when these bats go and forage on these big flowering pulses, they're no longer on this agricultural land. And so there's like really no opportunity for them to have a spillover. But kind of in other times, it does matter. I think the time spent over a paddock foraging is probably very predictive of pathogen spillover, right? But I don't think we've been able to quantify that well. Like actually getting enough replication with GPS tags and like watching bats forage, in the whole area is really difficult. But I think that's something they're actually working on right now. They've Manuel, the person who I talked about, and Reina actually have a study where they've got Doppler radar where they can actually watch like whole mass movements of bats, and you could look at how many bats are all over this horse paddock at this time, right? So you can sort of get an idea of just the time spent over there. But it does matter. But we don't directly have we don't directly understand that at the moment, but it's something they're studying. Yeah. Actually, I'm gonna zoom through this. I'm gonna skip this slide and just kind of give you basically how would you measure stress, right, in this population? Just to kind of give you some ideas. In this system, one of the things that they actually see is a good monitor of stress on a population level is how many bats actually show up at rehabilitation clinics. So they have wildlife rehabilitation clinics, where you see like birds with heat stress or something else show up, or with injuries, and they actually get help with recovery. So the number of bats that get intake into a rehabilitation clinic, those increase a lot during food shortages. So these bats are nutritionally stressed, and they're coming in sort of like in wasting condition. And it happens. It's very. I think it's true that a lot of them are juvenile bats, right? So these are bats who are young, who've not accumulated, enough fat reserves and sort of an individual kind of reserve of energy. So if there is a food shortage, they're disproportionately infected. The other thing that they monitor in the system is the reproductive success. So you can actually you can use just binoculars to look at whether a female actually has a pup on her. Huh? It's such a huge difference. So like when there is a food shortage, 50% of females may have a pup, but in years where there's not, they have good food, it's near 100% of females are able to give birth and successfully wean a pup. So this is one way of monitoring it. Monitoring like a stress and immune function, you can measure cortisol directly in blood using, there's multiple assays, immunoassays and radioimmunoassays. There's plenty of tests for it now. Alternatively, you can also kind of indirectly sample, so you don't have to capture an individual. You can also measure it in feces and urine, feathers, saliva, it's everywhere. Some measures for immune metrics that you might look at is like total white blood cell count, neutrophil to lymphocyte ratio, bacterial killing assays. There's a lot of options out there. Some are more interpretable than others. I think the neutrophil to lymphocyte ratio is somewhat hard to interpret sometimes, but it's a nice crude measure of just like an individual's stress response. And I will pass out my slides. You could use this one, but there's also, you might think of also like using metabolomics and proteomics and lipidomics to try to measure multiple kind of biomarkers of stress in a population. But yeah, some other things, total IgG, IgM, gene expression of like interleukins and other things might also tell you something about immune function. So these are worth kind of looking into if this is something that you want to measure. Ideally, you would want to measure stress, like cortisol levels, immune function, and pathogen shedding in the same individuals. So I'll leave it here. We just need more of this. We need more of this research to really try to make these connections because it's pretty rare in the literature. Ideally, we want good, well spatially replicated studies and longitudinal studies. Those are kind of like the ideal condition because longitudinal studies can really help us understand the causality, right? Is the pathogen a stressor, or is stress causing an increase in the pathogen?
So in your previous slide, you showed this non-invasive measurements of stress, right? Do we necessarily have to measure it from the urine from the animal, or can we take samples from the surrounding of the animal without even going to the,
I think that's the idea. Like that's the idea here is you might be able to collectively measure the amount of
environmental
cortisol.
Samples.
Yeah, if you can measure, say you take an environment, a gradient of degraded environments, like intact environments, degraded environments. You could measure at a population scale, like collect roost urine from across that gradient, and you could just say, are cortisol levels higher in the degraded environment versus the intact environment? So yeah, that would be sort of a non-invasive way of doing this. Yeah.
So, correct me, but so when you're looking for glucocorticoids, is this for basically measuring the acute stress?
Yeah.
So how do you figure out the baseline levels for a species like bats in the field?
I think it's important maybe to like do some initial sampling. And that's why longitudinal studies are really great for this, because you can actually measure sort of like the baseline level of stress, because a lot of things can affect cortisol levels. Like, there's actual DL cycles. You can have increased cortisol just at the end of the day because it's a hormone. It has an important function just in daily life. So you want to understand what those natural variations are. And maybe there's an increase slightly during pregnancy that you would perceive. But you want to know when it actually reaches a 5% outlier, right? That would be a measure of like really stressful conditions or a prolonged stress, right? Where you see cortisol levels persisting at a high level for a long time. So yeah, I think doing cross-sectional studies of cortisol is really tough, right, to interpret. So I think longitudinal studies are best for that.
wondering the purpose of horses. So is horse meat a thing in Australia, or this is just for leisure?
It's just for people riding. They have them as sort of status symbols, and yeah, there's like equestrian sport and everything there. You know, breeding horse stocks, but it's mostly for sport and recreation. Yeah, they don't eat it there. Well, thank you all for the questions. I hope this was interesting. And I can share this. The video will be online. Hopefully, I showed all the videos, all the slides. So, thank you.
Thank you, Cliff. Yeah.